Okay, we are recording now, okay?
So
we had 33 so far attending. So welcome everyone. Thank you for joining us. This is Virginia Women in HPC, and we are committing to fostering and supporting women engagement and equitable representation in high performance computing and all the other technological sectors. We are dedicated to enhancing diversity, equity, and inclusion. So we thank you for being here. To know more about us, this is a program that was actually founded in April of 2021. So far, we have 8 institutions participating: UVA, Virginia Tech, George Mason, VCU, ODU, William and Mary, Beans, and the University of Richmond. And today, we have a total of 341 members, a part of our mailing list. And our goal really is to support the inclusion of women and minorities in high-performance computing. We invite you to please use the QR code to visit our website, or you can just do the link that we have in the screen here. And with that, Ujita is going to be the moderator for this session today, and I will give you the platform. Thank you.
Thank you, Gladys, and welcome everybody. So today we are thrilled to introduce Dr. Daniel Ranfola, a true powerhouse in the world of data science and applied science. Dr. Ranfola wears many hats at William and Mary. He is an associate professor, the graduate director of applied science and data science, and the leader of one of the largest undergraduate research groups on campus called the GeoLab. But his passion doesn't stop there. He's currently pioneering exciting applications at the intersection of machine learning and satellite imagery analysis. Think beyond weather forecasts and into uncharted territory. Dr. Ranfola is constantly seeking bright minds to join his research journey. So if you're a Python whiz with a thirst for computer vision and satellite data, keep your ears perked. He might just be looking for you. A fun fact of Dr. Ranfola: with all his accomplishments, he sometimes forgets to update his CV, a testament to his relentless pursuit of knowledge. So please join me in giving a warm welcome to Dr. Daniel Ranfola.
Can you all hear me now?
Yes.
Yes, we hear you.
And can you see my slide deck?
Yes, we do.
Great. So apologies, first of all, I can't see any of you or see your questions. So if you have any questions, I guess we'll kind of chat at the end here.
I'll help with that, Dr. Ranfola.
Sorry, say it again.
I'll help with that. I'll keep an eye on the questions, and we'll take those questions after your presentation.
Perfect. Thank you. So yeah, I'm Dr. Dan Ranfola. I'm here at William and Mary. I've been here for about a decade now. Circa 2016, I founded and served as the inaugural director for our data science program. I'm a faculty member in applied science, and then I also do most of my service in the data science program. My background is largely in satellite imagery analysis. In modern days, that largely means a lot of deep learning. So using deep learning and computer vision techniques to extract information from satellite images. Today, I'm going to talk a lot about the different projects we use our high performance computing infrastructure for. So the types of kind of cool stuff you can do with satellite data, which is probably pretty far afield from the stuff that a lot of you are doing here. And then I'll talk a little tiny bit about like the types of infrastructure and whatnot that we use, but this will mostly focus on the applications. So my research, and by the way, when I say my research, I'll say at the very end, but you know, everything I'll be presenting here is not stuff I did individually. I have a hand in all of it, but my students, my collaborators, I mean, everyone has really contributed quite a lot. So the research in my lab is really focused on deep learning methodologies applied to 2 different pillars of activities. So a lot of work doing socio-demographic modeling and mapping, and then a lot of work associated with environment and climate change. So on the side of the environment and climate, there's really kind of 2 major activities that we work on. First is in collaboration with the Virginia Institute for Marine Science, VIMS. We've been doing a lot of work on mapping and modeling human structures with deep learning, as well as kind of marsh habitats. And then with the Global Environment Facility, which is the largest multinational donor of climate change aid, we've done a lot of climate change assessments. So to talk a little bit more and just kind of show you what this looks like. So in terms of kind of mapping marshland and shoreline structures, there's two different activities we've been doing a lot of work on. The first is from satellite imagery alone, to what degree can we distinguish between where marshes are and different types of marshes? So the communities within marsh types. We have a PhD student working collaboratively on that topic. Pretty wicked problem and a pretty important problem to solve. Very closely related to that, we have the shoreline structures database that we're working on. So very similar class of problem from satellite imagery. Can we identify where people have built these shoreline structures? Both of those seem relatively straightforward, but when we do this, we're doing this at scale, and we're largely doing it at with very, very high resolution imagery sources. So sub-meter imagery and across the entire Chesapeake Bay, right? Identifying where every single one of these shoreline structures is. So it turns into a big data and an HPC problem pretty quickly. Most of our work relies on GPUs, and so we do a lot of distributed data parallelization across our GPUs here at William & Mary to enable this research. This has really been pretty revolutionary. Prior to this technology, really the way that we would collect an inventory of shoreline structures would be manual. The most recent ones that we're aware of took nearly a decade to complete these inventories. And so, now we can do that in an hour using some of the deep learning methodologies that we've created. Not perfect, but pretty darn good. On the global scale for climate change, as I mentioned, we do a lot of impact assessment. So this is, many of you have probably heard of like the Paris Accords or the Kyoto Accords, these large international accords where we are promising to emit less carbon, or we are promising to protect a forest or plant more trees. The Global Environment Facility is the group that implements those. And what we do is we use time series records of satellite imagery of the entire world to establish the degree to which they're meeting their goals. So we work directly with the World Bank and the GEF to do this. It's a really, really big data problem on the satellite side. So we have a lot of satellite imagery of where these trees were, but of course, satellite images are not, they're pixels, they're not trees. So you have to do analysis of those pixels to identify how many trees there are within a given region. So we do a lot of HPC work, again, using GPUs primarily to assess those types of factors. On the sociodemographic side, so this is where we do, I think, some really neat and unusual stuff using the HPC. So two different tiers of activities, or two different types of activities here. First, we do a lot of mapping and prediction of sociodemographic factors. I'll talk more about that in just a second. And then we also maintain the largest database of geographic boundaries in the world right now here at William & Mary. So first, I'll talk a little bit about mapping sociodemographics. So much to many people's shock, in most of the world, the majority of the world, in fact, there are not censuses, right? We don't know where people live. We don't know the characteristics of those people. We don't know if they're educated, if they have access to healthcare, if they have high or low income. I mean, there's really very sparse information on people around the world. There are a few exceptions, right? Western Europe and the United States, and of course, a few Eastern countries. But for most countries, you simply don't know the status quo. That's a really big problem when it comes to things like distributing international aid. If you're going to, just as a simple example, you're going to fund a school to improve their ability to give education textbooks, right? As a pretty common example, what schools need those textbooks and which ones don't? In order to answer that question, you need to know where schools are and if they're already, functioning well or not. So what we've been exploring a lot is, to what degree can we estimate the quality of education or someone's income or other factors based on only satellite imagery? And the information we're exploiting here is correlations between the built environment and quality of a sociodemographic outcome. So the example that you see on the screen before you here, so on the left is the high school that I went to. So this is Chamblee High School down in Atlanta, Georgia. And you can see, right, it's a pretty large building. It has air conditioners on top of it. It has a paved parking lot outside. It has a big stadium next door. And then on the right is another high school that is from another county in Georgia. And you can see it's a much, much smaller establishment. The roofing structures are somewhat temporary. There are different materials. There's certainly no air conditioning on top. Some of the roads nearby are not paved. And so these kind of signals that we see in the data can help us kind of understand the resources that are available to these schools. It's not hard to figure out that the school on the left, Chamblee Charter High School, has more resources available to it than the school on the right, the West Sector High School. And so we can compare these two images and we can try to estimate which school is better resourced. And then we can use that to estimate the quality of the education that the students are likely to receive, and in turn, actually estimate their test scores. This is true not just in the United States, but also internationally. So this is a good example I like to use of two schools in the Philippines. And on the left, you see the school that you're looking at there. You can see the number on top of the building. That's very, very frequently associated with temporary structures that are put up during disasters. And so you can easily guess, right, that the school on the left is probably poorer resourced than the school on the right because they're relying on these kind of temporary structures. So this was with one of my PhD students now, Heather Baer. The study that we worked on relied on about 45,000 public elementary schools across the Philippines. So we had satellite imagery as well as Google Street View imagery of every single one of those schools. We know that we have school test score outcome for many of those schools. So we know what their grades were on standardized tests. And then we use all of that information to actually predict the school test scores. Turns out that works really well. So you can get pretty good estimates of their overall scores, and you can get pretty good overall estimates of if they're above or below average schools as well. And again, the cool thing here, that in my mind, the incredible thing here is that this is 100% only done on the basis of imagery. There is no other input information here. And so based on just the pictures of the schools alone, you can figure out if they're going to score above or below, the average student will score above or below average on an English test score with about 82% accuracy, which is pretty incredible. And we found this, by the way, to be true not only in the Philippines, but we've done multiple tests in lots of other countries as well, Brazil being the one that I remember off the top of my head. In addition to that, we also kind of extend that type of research into modeling outcomes. So one of the things that we do here is using data from Mexico in conjunction with the Department of Homeland Security is we do mapping and modeling of the number of migrants that are likely to leave a given location in Mexico. And we augment socioeconomic data with satellite imagery in this case. So we're really trying to pick up on signals that we wouldn't otherwise be able to see. Things like drought, right? When there's a drought, you can see that in a satellite image, and that helps to give you additional information that you can use for prediction, which is quite helpful. This works pretty well. Now, in this case, this is not satellite imagery alone. We do deep learning tasks and kind of modeling on a lot of different data sets to get kind of these tight correlations, but the satellite imagery adds to that quite a lot in these cases. With these as well, using the outputs that we generate from the high performance computing infrastructure, we set up models where you can interact with or websites where you can interact with the models on the front end to test different types of scenarios as well. So this is largely relying on kind of dynamic forward passes through the deep learning networks that we've developed. And right now, what we're working on is how we can expand this to other locations. So very, very GPU intensive, but effectively what we're doing is we're saying, okay, we know that we can use correlations between what households and what crops look like in Mexico to help us predict migratory flows. Can we do that in other countries? And the answer is probably yes, but unfortunately, or just the reality is different countries look different. The types of architecture they use is different. The types of crops they plant are different. And so we're trying to figure out how we can adapt the models that we have in Mexico to some of these other different locations. And in the deep learning community, that's a task called domain adaptation. And that's the focus of a lot of what Heather's working on today. In addition to that, on the HPC environment, one of the biggest challenges, as probably many of you on this call know, with deep learning is explainability. You know, like what are the actual features that we are keying in on these images? So the natural question, right, with let's say the schools example is, well, why? Like, what are the features in the schools that we're seeing that correlate between test score outcomes and the infrastructure? And I like to tell the story of like, okay, yeah, so the schools that have playgrounds, you know, we can see a playground from space, so they probably have better outcomes. But it's very hard to prove that, right? Like anecdotally, that may well be true, but actually finding techniques that allow us to prove that is in fact one of the features that it's keying in on is very, very tricky. So we've been doing a little bit of work on that in the context of satellite imagery. The next kind of socioeconomic thing that we do in the high performance compute world is one of the more bizarre things that we do here at William and Mary. So every single geographic unit of observation around the world has some sort of a footprint. So like if you live in Williamsburg has a legal boundary. If you live in a county in Virginia, like James City County, there's a legal boundary associated with that. There's a boundary of Virginia, right? We all know we live in the state of Virginia. Yet again, somewhat to my shock, most places around the world don't actually have agreed upon boundaries. So like imagine waking up and you're going to figure out, you need to get your license renewed, and you don't actually know if you live in Virginia or North Carolina, and there's no legal document that can tell you that. That's the reality in a lot of the world. And so we kind of fill that gap. We actually work with the World Bank, the Gates Foundation, the United Nations, and a number of other organizations to collect and distribute in a free and open way all of these boundaries. That doesn't sound like a high performance compute task on the face of it, right? I mean, no, it's just collecting data, but it's a huge volume of information. Every single time that a line changes directions, you have to record that. There's a point of data that tells you when a polygon is defined, where that line changed. And when you look across our full data set, you quickly end up in the terabytes of data. Every month, we generally are distributing and egress around 60 to 100 terabytes of information. We generally have between 750 and 1,000 users hitting our website every week, and that doesn't include our API. So this is a pretty popular data set. Just to give you an idea of the complexity of these things and why they can get so computationally intensive to build. So this is ADM1. This would be the equivalent in Malawi of like a state, the state of Virginia. And again, every single time any one of these lines moves, we have to have some point of data in our database that collects that. We also have to confirm that these shapes are topologically valid, that they actually make shapes and not like figure eights or something like that. And the math behind that is not trivial to compute as well. You can see as we get to ADM2, right, even higher levels of granularity, more and more data. And then when you get to ADM3, it really gets hyper detailed. So we end up collecting just a lot of information, a lot of maintenance there. This is a really fun task because we rely on, we curate the database. So we actually have professionals, including myself, that look at these files to make sure that they're in line with current legal standards within any given country. But the way that we kind of do this is through a combination of a online Azure-based pipeline in which people can basically submit a pull request to GitHub and automatically get spun up into a container to check the validity of the file. If that all works, then we pull that down into our local HPC, and there we do the full build. So that's a very intensive process that kind of requires us to do a lot of geometric manipulations across our full data set. Just for scale again, that full build on our HPC side using about 20 nodes with 20 cores each or so around 400 cores, you're generally talking about a 2 to 4 hour long build. One of the cool things, I wanted to throw this slide in because I know boundaries are kind of boring if you're not a geographer like me, but one thing I want you all to be aware of because it's cool. Depending on where you are in the world, so different people have different definitions of boundaries. So like the World Bank has some definitions, the United Nations has definitions, we have definitions, the Gates Foundation has definitions. Many of them are interrelated. Those groups use some of our boundaries and use some of their other boundaries. OpenStreetMap, Google Maps, right? Like all of these different sources have their own boundaries that come from different private companies mostly. But if you actually look across all these different sources, they disagree. So like if you want to know what is Virginia and you look at 3 different sources, they're all going to disagree what Virginia is. And that's a dramatic problem in different parts of the world. So in some places, if it's green on this map, they all kind of tend to agree. But if it's red on this map, they all tend to disagree. So if you're working in Russia and you want to do work that requires you to figure out where a given state is, you have to pay a lot of attention to the source you're using to define those types of states. And if you're working with census data or any kind of aggregated data, that can cause real problems. Well, that's it for me. So thank you guys. Again, I just wanted to reiterate, a lot of this work is done by the many PhD students and undergraduate students in my lab, and then a lot of wonderful collaborators at VIMS, NASA, Columbia, and a lot of other places. So thanks, guys. Happy to answer any questions.
Thank you, Dr. Ranfola. We have one question from Grace. Does the iffiness of boundary definitions not always apply to the definition of coastlines?
So coastlines matter quite a lot, and there are disagreements in what coastlines are because they both move over time and it depends on where you measure them. That said, coastlines are actually the least controversial part of what we do, because we can measure where they start and end using satellites at any given time. Where things get much more controversial is on the interior boundaries within countries. There's a lot, both across different countries, right? There's obvious disputes that everyone knows, like between India and China. There's a lot of disagreement about where those boundaries should be drawn. But also, like between James City County and Williamsburg in Virginia, there are disagreements about like where exactly water rights terminate and start. So there's a lot of minor disputes on internal boundaries that are very hard to resolve, because there's not a biophysical definition of like what the actual answer is. Whereas shorelines, at least there is a biophysical, like, oh, that is the shoreline.
Thank you. How many computation hours are consumed for the updates? This question is from Helen.
So generally we're talking around 1,600, give or take, for a full update across our data set. So we try to do nightly updates to it, because we get data fairly regularly from our community, and we're generally around 1,600 hours or so that we're running within a given night.
Okay.
Yeah, the actual problem we run into is not so much compute, it's memory. Because if you're trying to solve, is a polygon valid for Russia, and Russia has a definition of its polygon that has like 4 million vertices that define that polygon, the actual manipulations to solve for if it's reasonable or not require, you're talking probably like 32 to 64 gigs of memory on your node.
Okay. Carl, you want to go ahead and ask your question?
Sure. Hi, I'm Carl Friedrichs. I'm a professor at VIMS, and maybe Corinna, I see she's on the call too, might jump in. I'm assuming maybe, who are you working with at VIMS on the shorelines?
Corinna.
There, okay, perfect. And then this is very exciting, because one of the things I'm interested in is sources of sediment to Chesapeake Bay. And there's a lot of interest in the Chesapeake Bay program about how shoreline armoring is changing in time. And I'm guessing that with your work, because of course satellite photos are dated, we might get some answers to that, because it's like you said, was that the example of it took decades to map the shorelines, and now maybe with deep learning you can look at changes in time. So that's a really exciting application.
So
is there anything else you could say about what you're doing with that?
Yeah, so we literally, last Thursday, Corinna and I put in a proposal for a grant that would look at the impact of shoreline structures over time, specifically in this case on vegetative quality of marshland. So thinking about NDVI or EVI as proxies for that vegetative quality. The reason we went in that direction, despite NDVI having all sorts of limitations, is we can get the data, right? Like I can look at how NDVI has fluctuated over time. I think the sediment question would be fascinating, but I'm curious what historical data sets there are for, like Chesapeake Bay wide sediment loads, for example. And that's just a little outside of my scope of knowledge, because you can't measure that with a satellite.
Yes, there's this.
I can follow up also.
Oh, yes, please.
Hi, Carl. How you doing? First of all, also I want to acknowledge here Miranda LV. She's a dance PhD student. Also, she's a senior GIS analyst at the Center for Coastal Resource Management at VIMS. So she has been instrumental in all these process. So we have put that proposal with Dean and with Miranda and other colleagues in William and Mary. But also, just to go back to your question about the Chesapeake Bay program, we have been working very closely with Joseph and with Lou Linker from EPA, and we have been providing to them for this new iteration of the model, for the Chesapeake Bay model. They are actually using all the shoreline structure, the shoreline inventory for Virginia and Maryland. We just finished an EPA project for 3-year project, and we update all the shoreline inventory for Maryland, and so they are using that for the model. And we start now a new iteration for Virginia, starting October 1st with a DEQ project. And Miranda also is being part of that. And we are applying some of these techniques that actually Dan was showing today on this DEQ project. So hopefully by the end of this is a 3-year project, we will have also updated all the coastal counties in Virginia. So, and the Chesapeake Bay program is aware of that, and we're working together. So, yep.
That's very exciting. One of the trends in Chesapeake Bay water quality had seen a lot of places is that there's, of course, local exceptions, but the total suspended solids have tended to be going down with time. And the measurement of turbidity, which is how much particles reflect light in the water, has been going down. And one of the possibilities is that the increase in shoreline structures with time is reducing shoreline erosion and causing less sediment in the water. And the modeling that's been done by the Chesapeake Bay program for the open waters of the bay is that shoreline erosion is a larger source of fine sediment than the rivers themselves. So it's a really important issue, and it's very exciting for you to be using these techniques to look at that. And collaborating with great people at VIMS too. Awesome. Thank you.
Yeah. Thank you, Carl.
Thank you so much, everybody. This is such an engaging conversation. And thank you, Dr. Ranfola. Next, we have Dr. Priscilla Erickson. We have the pleasure of hearing from her. She's a true explorer of the natural world, both inside and outside the lab. Have you ever wondered how organisms adapt to changing environments? Well, Dr. Erickson delves into this fascinating question, studying the incredible world of tiny creatures with big implications for evolutions. Dr. Erickson joined the Berglund Lab in UVA in 2016 after completing her PhD at UC Berkeley, where she investigated the genetic secrets behind skeletal evolution in stickleback fish. Now her passion for genetics, evolution, and ecology leads her to study the amazing ways fruit flies adapt to seasonal changes. Today, Dr. Erickson will share her exciting research on evolution in real time with an invasive fruit fly. Get ready to witness the power of adaptation unfold right before your virtual eyes. Today, but Dr. Erickson's curiosity extends beyond the lab. When she's chasing answers, you might find her swimming, hiking, rock climbing, or tending to her native plant gardens, all with her trusty canine companion Mindy by her side. Talk about a well-rounded scientist. So please join me in giving a warm welcome to Dr. Priscilla Erickson.
Right, thank you so much. Let me just get my slides going here. The hardest part of giving an online talk is getting the slides going. All right, is that showing my title slide?
No, we don't see it yet.
Uh-oh. Oh, okay. How about now?
Yeah, we see it.
Okay, sorry, I didn't actually hit the share button. Here we go. Okay, I'm a little out of practice doing this Zoom talk thing. Fortunately, I'm glad to be out of the Zoom world most of the time. Thank you so much for that introduction. I'm really excited to tell you a little bit about the work that we're doing here at University of Richmond. And I added a subtitle to my talk, which is why I wish I hadn't put off learning to code for so long. So I'll talk about that a little bit as we move on. But as a biologist, I am really fascinated with the tremendous amount of biodiversity that we see in the natural world around us. So all around us, we have organisms that have diverse shapes and sizes, color patterns. They have unique adaptations that allow them to survive in some really surprising environments and interact with other species in various ways. And what I'm really interested in is understanding how all of these features evolve. How are they all encoded at a genetic level? Because at the end of the day, everything that an organism does is encoded in its DNA. And so I'm very curious how that works. So you might be wondering, I thought she was talking about fruit flies. Why is she showing me all these pretty animals? What I hope to convince you is that fruit flies are actually a really fascinating model to understand at sort of a very small scale how organisms evolve and adapt in changing environments. So one of the reasons that fruit flies are a really fantastic model for studying this is that they have a really, really short generation time for an animal. So an adult female fly can lay eggs, and about 10 days later, those eggs will be reproductively active adults. Or if you think about it, that means a fruit fly can be a grandparent when it's about 20 days old. So this is a really, really fast generation time. In addition to this fast generation time, they're also really easy to rear in the lab. So here I have a little vial of our flies in the lab. And I won't talk about it today, but because we can rear them in the lab, we can do lots of experimental work to complement some of the studies that we do both out in the natural world and using computers to study the genomes of these flies. Okay, because flies have this really short generation time, that means that changes in the environment can be really dramatic for a fly population. So if we think about fruit flies living out in an orchard in Virginia, the flies that are alive in the middle of the summer are going to encounter a very, very different environment than the flies that are alive just a few generations later in the winter. And so that means that we can actually see these fly populations evolving on sort of a human time scale. We can see changes in these populations as they adapt to different conditions because we have this rapid turnover of generations. And so my postdoc advisor at UVA, Alan Berglund, he did some really important work in this field where he studied a population of fruit flies living at an orchard in Pennsylvania. And he collected these flies in the spring and in the fall for 3 consecutive years. And he looked at their whole genomes and looked to see what was changing in the genome over the course of these seasons for these 3 years. And so he looked at lots and lots of different places in the genome and found that there were many different locations in the genome where particular genetic variants were changing in frequency over time. And you get this repeated pattern if you look across time. So we have this population in the spring and that has adapted to spring-like conditions. And then there's a number of genetic changes that occur as this population evolves and adapts to fall-like conditions. Then it changes back to that spring adaptation and back to the fall. And you see this repeated cycling. So this sort of parallel evolution. And this is evolution happening before our eyes because the most basic definition of evolution is that you have genetic changes in a population over time. That's what we have happening here. And so there's sort of this conception that evolution is this long, slow process that takes millions of years, but really it doesn't. It happens on the order of seasons. So all of this work was done in sort of the standard fruit fly that most people work on when they're studying flies. Flies are also a common model organism used for lots of genetic studies. The species is Drosophila melanogaster. And I previously worked on Drosophila melanogaster, but when I started at University of Richmond, I sort of wanted to take my work in a new direction, and I started working on a new species of fly. I think my species is a lot cuter than melanogaster because it has these cute little white stripes on its back. But it's also really interesting because it has a very different evolutionary history. So this is actually an introduced or an invasive species. It is from Africa originally, and it was introduced to South America by humans in about 1999. And then by human-mediated means, so produce transport and other sorts of things, it has spread out around the world, including North America. So it first showed up in Virginia in 2012 and now is quite common in Virginia and throughout the East Coast of the US. What's sort of interesting about these flies is historically, because they're from Africa, they're from a tropical region of Africa, so they haven't really ever historically encountered the seasonally varying environment that we have in Virginia. This is sort of a new challenge for them. And it turns out they're actually not very good at surviving here. So while this species is very common in Virginia in the summer when conditions are warm and humid, sort of the historical conditions this species has encountered, they don't do so well in the winter. And we find that they go almost completely extinct in the winter. So they are not very cold tolerant, and our cold winters here wipe them out. And so what this means is that actually we think that they're being reintroduced to Virginia each year from somewhere else. We think that they're probably established permanently in warmer parts of the US like Florida, and then humans are reintroducing them each year by moving produce around and other sorts of things. And so what we are using this for is as a model to understand what happens when organisms get introduced to new environments. How do they evolve? How do they adapt in those environments? And this is a really important question because introduced organisms can be really catastrophic for ecosystems and for humans. So living here in the South, you probably have driven along roadsides that were completely covered in this vine. This is kudzu, which completely takes over everything and kills all the other plants in its path. So these invasive species are often successful because they are able to evolve quickly and adapt to their new environment. So here we have a case where we have an invasive species that's repeatedly invading in the same environment year after year. And so we can study how is this invasive fruit fly evolving and adapting to new environments here in Virginia. So that's sort of the basic question that we're interested in. So why do I use high performance computing to answer this question? Well, we study this question at a genome-wide scale. So looking at all of the DNA that makes up the genome of the fly. And in fruit flies, the genome is about 150 million bases or chemical letters long. So these are the A's and T's and C's and G's that encode all the information it takes to make and be a fruit fly. So this is big to start off with. And to study it, we do what's called DNA sequencing. So we basically take fruit flies, we extract their DNA and chop it up into little pieces. And then we find out the orders of A's and T's and C's and G's in that sequence. And one experiment, we might derive hundreds of millions of sequencing reads or little pieces of DNA sequence, but then we have to do something with them. So we do a process called mapping where we use a reference genome. So this is sort of the known DNA sequence of a standard fruit fly. And we can align all these different sequencing reads we have to the different parts of the reference genome. And so this requires high performance computing because it uses a lot of cores and a lot of memory to work with all of this sequence data. Once we have the sequences aligned, we can actually find where are there genetic variants or genetic differences in the genome. So for example, some flies might have an A at a particular position in the genome and other flies might have G as their chemical letter. When we do this, we find there's about 5 million genetic variants in this 150 million bases of genome, and we have dozens of samples. So we're studying 5 million genetic variants across lots of different samples. And so this becomes a rather large computing question. So once we have these genetic variants, we can ask questions like what I showed you previously about how are these genetic variants changing over time? How do they vary across different locations that we've sampled, et cetera? An important thing that we do as part of this research is we can look and try to find trends in the data, but we want to know if those trends are real and driven by the environment. And so to do that, we use a permutation process. So we take the data that we actually have, and then we basically scramble it or randomize it, and then run the same analyses on scrambled data. And we'll do that thousands of times to sort of see what the null background expectation is so that we can see whether the patterns that we are observing are stronger than you would expect just by chance in scrambled data. So to do this, we're running all these analyses, but then we have to do them thousands of times over on the scrambled data. So we use Slurm array jobs to accomplish that. So all of this is why I had to learn to code. I am definitely trained as like a traditional biologist. I worked in the field, I worked in the lab. I avoided learning to code for kind of as long as I possibly could, but I ultimately realized that to do the sorts of studies and learn the types of things I wanted to learn, that this was the direction the field was going. Everything is using these high throughput sequencing approaches. And so I had to learn how to deal with the data. So I want to just tell you very briefly a few things that we've learned about evolution in this species. I'll try to keep it as straightforward as possible. One thing that's really interesting is that we've been studying these fly populations for about 6 or 7 years now. And one thing that we see is a signal of long-term genetic changes in the population. So the approach we use is called principal components analysis. The details don't really matter much, but basically we can get numbers that describe the overall genetic makeup of a particular fly population. Here we're looking at 2 different locations in Virginia, as well as a location in Florida, and calculating the statistic that sort of summarizes the overall genetic composition. And what we see is over time, from 2017 to 2022, we see all 3 of these populations changing in the same general direction. And so we think this might actually represent that these fly populations are evolving and adapting and genetically changing as they spend more and more time in North America and adapt to this particular environment. We did a more specific analysis to look to see whether the genetic changes that were happening in these populations were in fact similar. So this shows kind of a general trend, but we wanted to look at individual genetic variants and see if they were changing in the same way. And so this is where that permutation analysis comes into place. So we compared the genetic changes that we see over time in the 2 orchards in Virginia to one another and used the permutations to develop sort of a null background expectation of how similar we expect these populations to be. And we see that the actual populations are more similar in their changes than we expect based on the permutation. So the permutations predict that they should be about 50% similar, but in actuality, we see that they're about 58% similar. And so this suggests that all of these populations are in fact showing similar genetic changes over time, and they're actually more similar than we expect by chance. We can also look at evolution on even shorter timescales. So during a single growing season for these flies. So here I'm focusing on just a single genetic variant in the genome, and we're looking at how common is this genetic variant early in the year and late in the year. And you can see in all 6 years that we sampled here, the frequency of this genetic variant has increased over time. And so this suggests that these populations are all evolving in sort of the same direction in each year. So we're getting this repeated evolution after the invasion happens. So this is just a single genetic variant. We find that this happens in quite a few genetic variants throughout the genome. And once again, we can ask how similar are those changes if we look at multiple different orchards that we're studying in Virginia. So we did this analysis for one orchard in Virginia and a second orchard. And really surprisingly, what we find is that the two orchards, the way that their genomes are changing throughout the course of single seasons is actually less similar than we expect by chance. So again, we do that permutation analysis. The permutations predict that we should see about 50% similarity in these changes. The actual similarity across these two orchards is less similar than we expect. And so this suggests that the evolution that we're seeing might actually be very context dependent of where are these flies and what is the exact habitat and environment they're living in. The two orchards we study are fairly different in terms of location, management practices, et cetera. And so it seems like this is actually really important in influencing the short-term evolution of this particular species. So to kind of the take-home message is that these little flies can evolve really quickly, but sometimes that evolution can be a little unpredictable. But we're using them along with the HPC tools in our toolbox to study how invasive species evolve over time. So I'll stop there. I'm happy to take questions. I want to thank my funding from the NIH and the University of Richmond, as well as some of the folks who were involved in this. So Sam Morgan is an undergrad who did a lot of the DNA extractions for the samples I talked about. And Jerry He is a really fantastic student who did quite a bit of the analysis that I showed you today, as well as my technicians Logan and Weston, who actually sampled the flies in the field and kind of made all of this happen. So I'm happy to have people reach out if you have follow-up questions, and I can take questions now. Thank you so much.
Thank you, Dr. Erickson. Our first question is, how do you know whether the genetic changes are due to being in North America versus new populations coming in from other locations?
That is a really great question and something that we're still working on. So we kind of have this like hypothetical model that that we have this established population in Florida and they're coming in from Florida back to Virginia, but I actually have no concrete evidence that that is the case. And so the possibility that flies are being reintroduced from other species, from other locations is totally possible. And that's actually something that's quite common in invasive species that are getting moved around by humans. You can get populations, kind of like mixing in new and unexpected ways. So right now we don't have any evidence that there's anything like really, really different, like from a very different part of the world coming into Virginia, but it's certainly a possibility that we have to keep an eye out for.
Thank you. Gladys, we have a question.
Yes, I actually thank you, Priscilla, for your presentation. I really appreciate it. I actually have two questions. So the first one is more in terms of you talk about the genetic changes. What are did you observe specific gene families that are actually changing through the season that are triggering those changes?
That's a really good question. That is sort of like the next step to take this. Right now, we kind of just have a list of particular locations in the genome, but starting to find out like what are the genes involved, what are those genes potentially doing? That is a downstream question. I would say all of this is in still the very early stages.
Okay. And the second question is more in the technical aspects of the computational resources. I guess what I'm wondering is trying to understand, and this maybe help out the community thinking in terms of the memory demands that you had to do the permutations, and whether you encounter issues with trying to launch multiple arrays. And I imagine this was for the permutation, was not for the mapping, correct?
Right. The mapping is pretty straightforward. Like it sort of plugs through the permutations. Basically we just generate like 1,000 data files where we've reordered a particular column or something, and then launch basically the exact same code that analyze the real data, but to analyze the permuted data. That's how we typically go about it. And so it sort of runs through one job at a time. I don't know if that really answers your question, but.
Yeah, I guess, and your experience, right, trying to handling the data and the things that you learn about the coding, what has been your roadblocks in working
Oh, the
type of data?
I would say for me, my personal biggest roadblock is that I don't have a lot of formal training in this. So I'm kind of like flying by the seat of my pants most of the time and trying to figure out things as I go. I would say like computational wise, we haven't had any major roadblocks. Like the infrastructure that we have here has been like more than adequate to do the sorts of things that we want to do. I think training undergraduates to do this sort of thing has its own set of challenges. Jerry has done really fantastically. Sometimes there'll be like silly little mistakes that take a while to figure out. I think that probably happens to everyone who does programming. But I would say computationally, we haven't really hit any major roadblocks yet.
So memory has not been an issue and course accessible to you for the arrays? Yeah, or more or less, how much of the compute time have you been requesting
Oh
that has
gosh.
been for this?
Oh man. It has been a while since I submitted one of these jobs. I would say I don't have a good answer for that off the top of my head. I might have to look back into that and see. But yeah, I mean, sometimes it like does take a while to run everything. And I would say we haven't really done like a huge amount of optimization work to perhaps like there might be ways to do this more efficiently that we haven't done. Fortunately, we haven't had the pressure to do that because we have been able to do everything with the resources we have here. But if we were going to do this on a really, really large scale, we would probably need to think more seriously about optimizing. Sorry, I feel like that was not a good answer to your question. But...
That's completely okay. I understand. And my last question is more in terms of the tools. Have the tools been something that you have customized, that in-house scripts that you had to write, or you have specific tools that are, open source that you have used and be able to install in the systems?
That's a really great question. So we use a combination of open source like command line programs and open source R packages to do a lot of the manipulation. I would say the actual analysis, like looking for seasonal change or change over time, like those are more custom R scripts, but we're utilizing R packages that have been designed to work with the sort of data structures that we have for the like large scale genetic variants. So for the final stages, yes, customized, but almost everything else is open source.
Okay. Thank you so much, Priscilla.
Yeah.
Do we have any more questions? Feel free to post it or raise your hand and ask a question over here. All right. Thank you so much, Dr. Erickson. That was wonderful.
Well, thank you.
And as Gladys mentioned in the beginning, we will be posting you'll be receiving an email with a recording of today's presentations. Feel free to reach out if you have any questions, and keep engaging with us.
Yes, Urjita, I would like to mention to everyone that is here that we have, the way how we're going about our events, we are taking right now some feedback from the community. So anyone here that would like to place your comments in the chat, right? We are planning and we are trying to move towards to have an in-person event where we can actually do a highlight of lightning talks and poster presentations, allowing opportunities for the students from, or Virginia universities to actually come together into one place, create opportunities for in-person networking and also presentations. And so for those that are here that will be interested, if you can just add your information in the chat and whether would you be interested in something like that, we'll really appreciate it. Also, the committee, as I mentioned, we are institutions working together, and we are always looking forward to what are the topics that the community would like to be, listening, or things that would like to come out of the our program. So if you can add those to the chat, we'd really appreciate. Our next event is going to be announced in a couple of weeks. It's going to be save the day for April, and we're still deciding in the direction we're going to go for that. But if you have any comments, we are willing to take the feedback. Please, if you have time, visit our website. I put it in the chat. It's our VA-WHPC.org. And you're going to see that if you're interested in seeing past presentations of the lightning talks that we have hosted for our students, they're also going to be recorded there. And with that, I just want to say thank you for your time. Thank you for visiting us. And please leave your comments. We really appreciate the feedback. Thank you. I'm going to stop the recording now.